So another crop is okra. So it is called gumbo in North America. Okra was originated in, actually there was a contradiction regarding the origin of the okra. And generally three places they say. One is Ethiopia, another one is India, another one is West Africa, especially Ghana, as the origin of the okra. Okra is a polyploid species. Epilomascus tuberculatus n is equal to 29, and another unknown species n is equal to 36. Natural hybridization was occurred, and followed by the F1 had the natural doubling of the chromosomes. So thus the cultivated Epilomascus esculentus was originated with a 2n is equal to 130. And there are three cultivated species in okra: Epilomascus esculentus, Manihot, and Mostatus. Epilomascus manihot was also used as a leafy vegetable in Africa, and it is resistant to yellow vein mosaic virus. And there are actually there are four resistant species identified against the yellow vein mosaic virus. We all know that yellow vein mosaic virus is a major disease in okra. So this manihot subspecies, manihot and Tetrophilus, all these three species can be hybridized with the cultivated Epilomascus esculentus. But the problem is all these three species do not have the true resistance. They have false resistance only. That means they have the infection of the virus, but they do not exhibit any symptoms. That means they are the symptomless species. So when we hybridize, when we transfer the resistance from this manihot subspecies, manihot and Tetrophilus to the cultivated types, the resultant progeny is generally resistant for few years only, 3 years or 4 years. Afterwards, eventually they are breaking the resistance because of the false resistance in the source. That's why Pusa Savani or Arkanamika or Kabai, Punjab Padmini, Parbani Kranti, lot of YVMV resistant varieties were developed by using these three species as a source of resistance, but eventually they lost the resistance due to lack of true resistance in the source, at the same time developing of the new variants in the virus also. But there is one species, Epilomascus pungens, which is having the true resistance to the virus, but it is not possible with the cultivated Epilomascus esculentus. That is the problem. There is a hybridization barrier between these two species. And Epilomascus ankylosus is resistant to frost and low temperature. This is also one of the most commonly asked questions, Epilomascus ankylosus. And there are some other species like Ficalnius, Cnitus, and LA. And Epilomascus esculentus okra is rich in iodine content. It is used against the So YVMV is an important disease, as we talked earlier, and it is transmitted by the whitefly. The resistance is bound by the two complementary dominant genes, and okra is an often cross-pollinated crop. And for export quality okra, the fruit length should be 6 to 9 centimeter length. This is also previous question. So the export quality okra should have a fruit length of 6 to 9 centimeter length. So backcross and pedigree breeding method was the adopted breeding method for the YVMV resistance breeding in okra. So Parbani Kranti was developed. Backcross pedigree means the F1 was hybridized to the recurrent parent for 3 to 4 generations. After 4 generations of backcrossing, pedigree method is followed. I think you know how the pedigree method goes. So just go to textbooks and just read what is pedigree, what is backcross. So the backcross pedigree method was followed in okra for transferring the YVMV resistance in Parbani Kranti and Punjab Padmini, P7, Arkanamika, Kabai. MDU1 is a gamma irradiated mutant of Pusa Savani developed by Tamil Nadu Agriculture University. And Punjab 8 or EMS8 is an EMS induced mutant. EMS means it is a chemical mutagenic agent, ethyl methane sulfonate. It was developed by Punjab Agriculture University, and there were some important varieties released by IIBR also. Just go to the website and see. And there were some good hybrids that were developed by the private companies which were popular. Excuse me. They are Varsha and Vijay by Indo-American Hybrid Seeds, Organic and Panchali by Century Seeds. Based on the processing purpose, for the processing purpose, there are some suitable varieties. For canning, Pusa Savani, Janardhan, and for dehydration, Pusa Savani selection 2, for freezing selection 2. And Perkins Green Lawn is an introduced variety. So potato, among all the tuber crops, potato is the temperate one. Remaining other tuber crops are the tropical types. So potato was originated in Peru, Bolivia, and in South America. And potato was introduced to India in 17th century. So the diploid species are Solanum furegia and Solanum stenotonum. We know stenotonum is a diploid one, it is a progenitor. So the diploid Solanum stenotonum was hybridized with some other unknown species, and the F1 had the doubling of chromosomes. So that the cultivated Solanum tuberosum, subspecies tuberosum, and subspecies andigena ramga. What is that one? Actually, here in potato and sweet potato, it is not exactly known whether they are the autotetraploids or allotetraploids. Allotetraploid means that what I said earlier, hybridizing between two species, and the F1 had the doubling of chromosomes. That is the allotetraploid. Autotetraploid means without hybridization, the diploid species itself will have the doubling of the chromosomes naturally. That is autotetraploid. But in the different books, they are reporting differently, especially in potato and sweet potato. Some are saying it is an autotetraploid, and some are saying it is an allotetraploid. But anyhow, it is a polyploid. There are triploid species, Solanum demisum, and pentaploid species, Chaucha, Zizpesky, and Cactylogum. The diploid Solanum species are the gametophytic, had the gametophytic self-incompatibility, whereas the tetraploids and heptaploids are self-compatibles. And the subspecies Andigena produces tubers in short day conditions in hills, and whereas the subspecies Tuberosum produces tubers in short day conditions in tropics, long day conditions in hilly areas. And long days promote the hulm growth and no tubers. Yes, for tuberization of potato, especially in India, that we have subspecies Tuberosum, requires short days for tuber production and long days for flower production. So the potato in India do not produce tubers under long day conditions. Hull means the above ground portion. The above ground portion grow very well in long day conditions, but it will not produce any tubers. For tuber production, the subspecies Tuberosum requires short day conditions. For flowering, it requires long day conditions and low temperature. So that the potato breeding station is located in Kufri in Himachal Pradesh, because in summer season there will be long days, at the same time low temperature. So the potato flowers in those conditions, long day conditions plus low temperature. So the potato breeding station in India is located in Kufri, Himachal Pradesh. So sometimes in plains also we can induce the flowering of the potato by extending the day length during winter season, by supplementing the light with artificial lights. So there is one more technique, true potato seed. Generally potato is propagated through tubers. Why can't with seeds? Yeah, potato can also be propagated through seeds, but the disadvantage of true potato seed is we need nursery. First we have to raise a nursery, and we have to transplant them into the main field. And in first season it produces very small tubers. That small tubers again we have to plant in the second season, then it produces a large size tubers. That means propagation through seed takes 2 years, 2 seasons, and it is very labor intensive because of the nursery and so many transplantings. So the cultivation of commercial potato crop through true potato seed is not at all economical. And the true potato seed is having the dormancy for 5 to 6 months, and the dormancy can be broken by treating with the GA3 1500 to 2000 ppm. And earthing up is an important agricultural operation in potato. 1 and 2 months after planting, we have to earth up the plants, because after tuber formation, if we do not do earthing up, the tubers will be exposed to sunlight. When the tubers are exposed to sunlight, chlorophyll pigment will develop that causes greening, which is a physiological disorder. And the greening is responsible for the development of alkaloid solanine in the potato tubers. That means, you just remember, don't confuse here. Greening is due to exposure to sunlight and development of chlorophyll. Greening is due to chlorophyll. This greening is responsible for the increasing of solanine content. If solanine is more than 20 milligrams, it will become toxic. So solanine is not responsible for greening. Greening is due to chlorophyll, due to exposure to sunlight. Because of the greening, solanine will form. So to prevent greening, hulm cutting, this is also very important operation in interculture operation in potato. 15 to 20 days before harvest, we have to cut the Hulms. Hulms means the above ground growing portions, the leaves and the shoots. We have to cut them 15 to 20 days before harvest. Generally, why we recommend hulm cutting before harvesting means uh, potato is generally is grown in winter season in the North India, generally planted in October and November, and the harvesting will be in January and February. So by the end of February, when the temperatures rise, the aphid population will increase. The aphids are the vectors of the viral diseases in potato. So when at the end of cropping season, the time of harvesting, when the temperature rises, the aphids will come, and the aphids spread the viral diseases. So to prevent the spread of the viral diseases through aphids, we cut the haulms when the temperature rises at the end of the cropping period, just before the harvesting. So when you cut the haulms, when you cut the shoots and leaves, there will not be any aphid infestation, isn't it? So there is a chemical, copper sulfate, that is used. That is also used instead of cutting manually the haulms. You can spray the copper sulfate. It will kill the above ground growth portions. And there are some important varieties and crosses you can read from any textbook. And Kufri Swarna is resistant to lead blight and cyst nematode. Kufri Sheetman, Kufri Deva tolerant to frost. Kufri Sheetman is tolerant to drought also. And Kufri Surya, this is a very important question. In the last 2 to 3 years, this question is appearing every year. Kufri Surya is a high temperature tolerant variety. It can produce tubers at about 25 degrees centigrade. So remember Surya means sun, isn't it? So, so it can be grown under high temperature conditions. Just to remember Surya. Surya means sun, automatically high temperature variety. And red skinned ones are the Kufri Red, Kufri, Sinduri, Kanchan, Lalima, and Aruna. Seed plot technique was developed by Dr. Pushkarnath. ETL for aphids. 20 aphids per 100 leaves is not much problematic, but if the number of aphids is more than 20 per 100 leaves, we have to go for the plant correction measures. And the tubers have dormancy. The tuber dormancy can be broken by 1% thiourea plus 1 ppm GA for 1 hour. And here is the important one. AMA1 gene. AMA1 is Amaranthus albumin 1 gene. This is an Amaranthus gene, and this gene is responsible for the high albumin and protein content. And this gene was transferred through recombinant DNA technology from Amaranthus to potato, from the Amaranthus species, Amaranthus hippochondriacus. The Amaranthus hippochondriacus is having the gene AMA1, Amaranthus albumin 1. This gene was transferred to potato through recombinant DNA technology for increased protein content in the tubers. This is also one of the important questions. So just these are the recently released varieties of potato. They have been released maybe 3 to 4 years, just 3 to 4 years before this, maybe between 2016 and 2020. So these are the variety Kufri Nilakant. It is a purple color variety with shallow eyes and yellow flesh. The skin color is purple and the flesh color is yellow. Kufri Nilakant. Kufri Manik, this is a biofortified variety with red skin color and yellow flesh tuber. We know Kufri Chipsona 1, 2, 3, they are the chip varieties, and this is the French fry variety, Kufri Fryon. And Kufri Chipsona 4, for processing, potato tubers should have high dry matter content. The dry matter content in the potato tubers should be more than 20% for processing. What happens if dry matter content is less and sugar contents are more? When sugars are more, generally by making the chips and the French fries, we have to fry the potato slices. When you fry the potato slices and the sugar content is more, there will be charring of sugars, melanin content will develop. You might have seen in local made potato chips, sometimes there is a black color mark. In local, in Lays you cannot find. In Lays they take all care, but in locally made potato chips, sometimes you can find black color discoloration. That is due to charring of sugars. That means the sugars burn when you fry the potato slices. If sugars are more, they burn and they produce that black color thing. So that for processing purpose, the potato tubers should have dry matter content more than 20%, and they should have less sugar content. And Kufri Sangam, this is GL purpose, means for table purpose and processing purpose. And this is an important variety, Kufri Thor 1, 2, 3. They are water use efficient potato varieties. Water use efficient means they can tolerate moisture, stress conditions. They can tolerate drought conditions. They can grow with limited available water. So if we tar 1, 2, 3, remember recently released varieties drought tolerant ones. Tar, there is a tar desert in Rajasthan. Desert means no rain area, very less rain area, like less water available area. So remember like that. Kufri, tar, 1, 2, 3. These three are the drought resistant varieties of potato released recently.